The genes you don't get from your parents, but can't live without. Inside our cell, each cell has a second set of genes completely separate from the 20 pairs of chromosomes we need from our parents. And this isn't just the case for humans. It's true of every animal, plant, and fungus, but nearly every multicellular organism on Earth. This second genome belongs to our mitochondria, an organelle inside our cells. They're not fully a part of us, but they are separate either. So why is so different from anything else in our bodies? Approximately 1.5 billion years ago, scientists think a single-celled organism engulfed the mitochondria's ancestry, creating the processor of all multicellular organisms. Mitochondria play an essential role. They convert energy from the food we eat and oxygen we breathe into a form of energy our cells can use, which is molecules called ATP. Without this energy, our cells start to die. Humans have over 200 types of cells, and all except mature red blood cells have a mitochondria. That's because red blood cell job is to transport oxygen, which mitochondria would use up before it could reach its destination. So, all mitochondria use oxygen and mitochondria's energy and have their own DNA. But mitochondrial DNA varies more across a species than other DNA. In mammals, mitochondria usually have 37 genes. In some plants, like cucumbers, mitochondria have up to 65 genes, and some fungal mitochondria have only one. A few microbes that live in an oxygen-free environment seems to be on the way to losing their mitochondria entirely. And one group, oxymonosaccharomatoids, already have. This variety exists because mitochondria are still evolving both in tandem with organisms that contain them and separately on their own timeline. To understand how this is possible, it helps to take a closer look at what the mitochondria inside us are doing, starting from the moment we are conceived. In almost all species, mitochondrial DNA is passed down from all one parent. In humans and animals, the parent and the mother. Sperm contain approximately 50-70 mitochondria in the tail to help the swim. This differs with the tail after conception. Meanwhile, the egg contains thousands of mitochondria. Each contains multiple copies of the mitochondrial DNA. This translates over 150,000 copies of mitochondrial DNA that we inherit from our mothers, each with independence and could vary slightly from the others. As a fertilized egg growing and divides, those thousands of mitochondria are divided up into the cells of the developing embryo. By the time you have different tissue and organs, variation in the mitochondrial DNA are just scattered at random throughout our bodies. To make matters more complex, mitochondria have a separate replication process from our cells. So, as our cell replicates dividing, mitochondria end up in new cells, and all the while they're fusing, dividing themselves on their own timeline. As mitochondria combine and separate, there's a cast of faulty DNA or mitochondria that aren't working properly for removal. All this means that random selection of a mother's mitochondrial DNA you will inherit at birth can change throughout your lives and throughout your body. So mitochondria are dynamic and to degree independent, but also shaped by the environment, us. We think that long ago some of their genes are transferred to the host genus. So today, although mitochondria have their own genes and operate separate from the cells that contain them, they can't do this without instruction from our DNA. And those mitochondrial DNA is inherited from one parent. The genes involved in building and regulating mitochondria come from both. Mitochondria contains defined tight classification. Their storage is still unfolding inside of each of our cells, simultaneously separated and inseparable from our own. Learning more about can give us tools to protect human health in the future and teach us more about our history. want more with slow speed. Inside our cells, each of us have a second set of the genes completely separate from the 23 pairs of chromosomes we inherit from our parents. And this isn't just the case for humans. It's true of every animal, plant, and fungus, and nearly every multicellular organism on Earth. This second genome belongs to our mitochondria, an organelle inside our cell. They're not fully a part of us, but they're not separate either. So why are they so different from anything else in our body? Approximately 1.5 billion years ago, scientists think a single-cell organism engulfed in the mitochondria's ancestry created the procedures of all multicellular organisms. Mitochondria play an essential role. They convert energy from the food we eat and oxygen we breathe into a form of energy we cells can use, which is molecules called ATP. Without this energy, our cells start to die. Humans have over 200 types of cells, and all, except mature red blood cells, have mitochondria. That's because red blood cells' job is to transport oxygen, which mitochondria would use up before it could reach this destination. So, all mitochondria use oxygen and metabolism to create energy and have their own DNA. But mitochondria's DNA varies more across species than other DNA. In mammals, mitochondria usually have a 37 gene. In some planets like cucumbers, mitochondria have up to 65 genes, and some fungal mitochondria have only one. A few mitochondria live in oxygen-poor environments seems to be the only way to losing their mitochondria entirely. And one group, oxymonanes monosaccharidoids, already have. This variety exists because mitochondria are still evolving, both in tandem with the organisms that contain them and separately on their own timeline. To understand how they possible, it helps to take a closer look at what the mitochondria inside us are doing, starting from the moment we conceived. In almost all species, mitochondrial DNA is passed down from only one parent. In humans and most animals, that parent is the mother. Sperm contain approximately 50 to 70 mitochondria in a tail to help them smell. These dissolved with their tail after conception. Meanwhile, an egg contains thousands of mitochondria, each contain multiple copies of the mitochondrial DNA. This translates to over 150,000 copies of mitochondrial DNA that we inherit from my mother, which was independent and could vary slightly from the other. As a fertilized egg grows and divides, those thousands of mitochondria are divided up into the cell of the developing embryo. By the time we have different tissues and organs, variations of the mitochondrial DNA are scattered at random throughout our bodies. To make matters even more complex, mitochondria have a separate replication process from our cells. So, as our cells replicating by dividing, mitochondria end up in the new cells, and all the while they are fusing and dividing themselves on their own timeline. As mitochondria contain and separate, they scatter virtually DNA or mitochondria that is working properly for moving. All this means that the random selection of your mother's mitochondrial DNA you inherit at birth can change throughout your life. and throughout your body. So mitochondria are dynamic and to degree independent, but they're also shaped by their environment, us. We think that more ago some of their genes are transferred to their host genus. So today, although mitochondria have their own genome, they replicate separate from the cells that contain them. They can do this without instruction from our DNA. And though mitochondrial DNA is inherited from one parent, the genes involved in building and regulating the mitochondria come from both. Mitochondria contain to define tight classification. Their story is still unfolding inside of each of our cells, simultaneously separate and inseparable from our own. Learning more about it can both give us tools that protect human health in the future and teach us more about our history.